So I get started. I first did my undergraduate plant identification course, and yeah, it's. I don't know. Did you get the bug in that course, or do you already have the bug?
I already had it.
Yeah, a little bit already had it, but anyway, it's pretty obvious it's going to be a good student. And that is kind of the obvious point out a mistake in the exam equipment. But he got his own back because he stole my office. I turned up to work one day. It's like, you've got to get out. Patrick's going to be moving in. So there you go. Patrick's did his honours with me on describing a new endemic grass species which grows around springs in central Queensland. We call it circumfontanales. I think you covered that. Good name. Around the spring. And then he bailed out, went to Melbourne and worked on eucalypt genetics, the grey box group, which anyone knows anything about eucalypts, they're represented around here, Eucalyptus norcana. And spreading his, and they've come back to Queensland and stole my office, got the job as a conservation geneticist at the Herbarium, which is fantastic. Look forward to him developing collaborations with students at UQ. And he's obviously, and this is what I'll be excited to learn about, is now spreading his wings and thinking more generally about what genetics means for conservation, the subject of the day. Welcome, Patrick.
Thank you, Rod. Creatively or not, part of the presentation today, the use of genetic data to inform conservation management of flora. Basically, I'm going to tell a little bit of story here, how we use genetic data in the government sphere to manage threatened plants and ecosystem and land management, that sort of thing. Yeah, so I'm with the Queensland Herbarium, which is part of the Department of the Environment, Tourism, Science and Innovation, but also have an honorary fellowship with Coffee here at UQ. We start with Northern Country, both the Turrbal and Yagara people and whose land we meet today and we work on, but also the many, many different people groups whose lands, sorts of lands I'm going to be talking about here today. We'll be covering a lot of the East Coast, so many different people groups. Yeah, not to repeat what Rod's already said, but I'm familiar with a lot of you here in the room. I thought I'd just give a bit of an overview of my journey to where I am currently. So I was a UQ undergrad, did my undergrad and honours here, finished honours, I think we decided in 2017 with Rod and then Cook in what was School of Biological Science, I don't know if that name's changed. And as Rod said, outcome of that was description and population survey of the Artesian spring endemic grass, Queensland. For some reason, I only have a black and white photo of that in there. Then went down to the University of Melbourne to do a PhD, got out of Brisbane for a bit. That was moving from grasses to eucalypts, so I know how to pick those easy groups to work in. Also was down in Melbourne through those lovely Victorian COVID lockdowns, which made writing papers really easy because there was not much else to do. So a lot of that work was using, sort of bridging that gap between phylogenetics and population genetics to look at biogeography and evolutionary patterns across southeastern Australia. When I finished PhD, got hired, was actually hired through Coffee here at UQ, but that was just formality. It's actually working down at the Royal Botanic Gardens of Sydney at the Research Centre for Ecosystem Resilience, doing a lot of restoration genetics studies. So that's, we'll come to what restoration genetics is, but that basically was going from academia to very much governmental focused work, very governmental science, where what we were producing was these big reports to stakeholders, land managers, giving them advice on how to manage their land, how to go about restoration, considering genetic diversity, genetic patterns and that sort of thing. So that was a bit jarring going from academia to government science where, you know, publication is something you do if you have a spare minute. We have hundreds of data sets that have never seen the light of day because they were handed to the relevant stakeholder and you move on to the next thing. And then in mid last year, so June last year, joined the Queensland Herbarium as their full-time conservation geneticist, set up a conservation genetics program here in Queensland, mainly looking at flora, but not necessarily limited to flora. Fungi too, maybe not animals. see about that. Doing very similar things to what we're doing down in New South Wales, producing these reports to land managers, best to go out managing their species, in this case, mainly threatened species. Given genetic knowledge we can generate. So, it's a bit of background. Now, background on the actual science. Just thought I might go through some of these terms I'm using. So, what are all these genetics? Population genetics, most people are probably familiar with what that is. That's using genetic data to study population dynamics across the landscape, things like patterns of genetic diversity, gene flow, rotations of populations, that sort of thing. Conservation genetics is just using those same tools from population genetics to inform conservation and land management, that sort of thing. And then restoration genetics is sort of a subfield, restoration genetics, where you're looking at, rather than threat species, we're looking at maintaining genetic diversity in the landscape of all species, so in those common species. Sort of as the talk goes, comes to why that's an important thing to consider. A lot of times it's possible to do more harm than good if you're doing large-scale plantings. So these are just some examples of the sort of analysis we're doing. So we're doing sort of cluster analysis of what is the structuring of genetic data between population across the landscape. Things like what is the relationship between genetic divergence of populations and how far apart they are in the landscape, so it's isolation distance. And then things like measuring genetic diversity of individual populations, comparing that, how it fits across the landscape. So yeah, they're the basic tools using to do all this work. Get a bit more complicated, but yeah, for a baseline. So why do we do it? Well, basically, if you don't understand what the existing genetic patterns are, what the evolutionary relationships are, the reproductive biology of species, you're sort of blindfolding yourself when you're trying to do any sort of management of them. These are key factors that influence how those species survive, exist, and form part of ecosystems. And it's a very powerful tool that nowadays is very easy to apply, which covers a very wide range of questions you might be interested in about any given species. So I've sort of broken down into 4 key areas, which are going to form the bulk of the talk today. First, that is, we need to know what we are conserving. Taxonomy is a hypothesis. It's not always correct. Plant species can have complex evolutionary history and genetic processes that you can't pick up just by looking at its ecology, its morphology, population dynamics. You can pick up a lot more about understanding what that species is doing, actually looking at what the population genetics are. Obviously, understanding a species' reproductive strategy and genetic patterns of genetic diversity can really inform how you go about managing that particular species, and how to make sure you're doing good rather than harm. And basically, because genetic data is a tool, really just empowers us to be more cost effective, which in government is a key concern. In how we go about managing these, we can make sure we're targeting our activities to where they're going to have the most positive impact for the least price. Yeah, that sort of thing. So yeah, as I said, data availability is growing exponentially in the genetic data sphere. So personally, in the last 5 or so years, I've analysed data for more than 100 species at the population level. That represents more than 1,000 collection sites, primarily across New South Wales when I'm working down there, but growing within Queensland. Biggest data set we had was more than 2,000 individual wild plants, genotyped, patterns for them. So these are all the species I worked on. You can see there's very much a taxonomic bias in there. My eucalypts, but they're ecologically important. So good place to start. I tried not to put too many examples of eucalypts in here. Tried to keep it varied, but we'll see. Yeah, so going to that first point, we need to know what we're conserving when we're actually undertaking conservation. As I said, taxonomy is a hypothesis, and genetic data is a very powerful tool to test it. Consider traditional taxonomies based on morphological characters. You have a small set of characters you're looking at. Basically, you can treat each position in the genome as an individual character. So we're talking about data sets here of 40,000 characters, where traditionally we're using 100 characters or something. Just that level of data is hugely increased. Allows us to understand that relationships much better. Traditional systematics relies on phylogeny, so that's phylogenetics. So looking at bifurcating relationships, you know, what did these things diverge from one another? How long ago was that? That sort of thing. Works well for lots of species, especially when you're talking about older things. Once you get to the recently diverged things, your typical phylogenies sort of falls apart, especially in plants where a lot of relationships are not actually bifurcating, a lot of reticulation, a lot of ongoing gene flow, that sort of things. So that's where population genetics can come in and be a very powerful tool to actually understand what are the dynamics occurring on the landscape in present day that can then inform how we classify organisms. Because you ask a species, there's no such thing as a species, but we need something to be able to actually manage it. So yeah, just some examples of where taxonomic resolution has been gained through these conservation genetic studies we've done. So this is looking at Pulmonea, so a P. Pulmonea maritima is a vulnerable, well, still is, it won't be for long, vulnerable headland shrub from New South Wales, occurs on 14 headlands up the coast. Very morphologically similar to the much more common Pulmonea villosa, which goes not on headlands. Turns out when you look at the genetic data, they're indistinguishable from one another, and the morphological difference is purely just because it's growing on an exposed headland. So not a valid taxon. Pulmonea villosa is not threatened, so Pulmonea maritima should definitely not be threatened either. But in this case, we actually find an interesting pattern here. So this is genetic clustering across, basically this is ordered down the coast here of the sampling sites. So red sites here, Pulmonea maritima sampling sites, yellow Pulmonea villosa sampling sites. You can see we have a lovely genetic break here somewhere in the mid-north coast of New South Wales that actually corresponds to 2 species. So we had the right number of species, it just didn't have the right circumscription for those species. And yeah, neither of these can be threatened. They're not uncommon. That's pretty good conservation outcome. We've decreased the number of threatened species by 1. Too easy. This is a very similar example on eucalypts. Eucalyptus rubida, there was basically 3 species in this group. Eucalyptus rubida has 2 subspecies in New South Wales. Subspecies rubida down southern tablelands. Subspecies barbigorum, which I think means from the land of the bearded people or something, up in the northern tablelands, and Eucalyptus canovalensis just from Mount Canovalis near Orange. Then you have Eucalyptus dalarumpleana, subspecies Heptantha up north, subspecies dalarumpleana down south. You look at them from a genetic standpoint, you find actually we can only find one genetic lineage up in the northern tablelands, and what's previously been called rubida subspecies barbigorum, it's the same thing as dalarumpleana subspecies Heptantha, which fits with the surveys they did by the consultants going out looking at those threatened species who said we can't recognise them in the field. So there's your first hint that something's wrong with the taxonomy. And we also found that Eucalyptus canovalensis really do separate from Eucalyptus rubida subspecies rubida. Great. And we also find the northern tableland stuff's most similar to dalarumpleana subspecies dalarumpleana from down south. Fit with that species. We then published taxonomic revision of the group. Get rid of rubida from the northern tablelands. It's never there in the first place. Everything in the northern tablelands now dalarumpleana subspecies Heptantha. That's not threatened. It's pretty common up there. Eucalyptus canovalensis becomes Eucalyptus rubida subspecies canovalensis because it is morphologically and ecologically distinct. So wanted to maintain it as a taxon, even if we can't separate it genetically. So that stays as a threatened species or threatened subspecies. So again, we've reduced the number of threatened species by 1. Nice. Very easy to do. But yeah, this is just an example to say it's not all one direction. It does go the other way as well. So Divisia ulicifolia is a little spiny pea, horrible thing to collect tissue samples from. Very spiky. Currently, there's 6 recognised and morphologically identifiable subspecies. We're only looking at 4 in New South Wales, the other 2 are more in South Australia. So we're included in the study. We actually find that there are 4 very distinct species here. These are distinct at the level of species from a genetic standpoint. Some of these correspond very well to the existing subspecies. So we got subspecies Stenophylla coming out really distinct here. Subspecies ulicifolia is up here as distinct. 2 of them go together, Piligensis and Rustifolia, don't separate genetically, so they probably are the same thing. Then we have this entirely novel lineage that doesn't, morphologically, it seems to be subspecies Ulicifolia, similar to that, but genetically it's very distinct. And then we have one site where we have some hybrids between those lineages. They're all perfect F1 hybrids, which is further evidence that we're dealing with species here, not subspecies or something. There's no gene flow between them. If you look at the distribution, they co-occur all throughout Eastern New South Wales. There's no evidence of gene flow between them. You can't really call them the same thing, can you? So yeah, work is underway to actually do that taxonomic provision. None of those will be threatened. They're all relatively common. But yeah, game does go both ways. All right, moving on to that second point of why we do this sort of work. Plant species can have complex evolution histories, and you can't actually see that by going looking at in the landscape a lot of the time. So organization is frequent between plant species. I won't really touch on that here, but yeah, is what it is. Does cause major conservation concerns, especially when you've got a naturalized species hybridizing a native species. But yeah, patterns of gene flow can vary greatly between species, even when they form part of the same ecological community. Done some work within woodlands. Found that some species you get near continuous gene flow over 100 kilometers. Others you find is really heavily structured. Same ecological community. You wouldn't pick that up without genetic data. Structuring is not always what you'd expect, given biogeographical patterns and biogeographical barriers. Sometimes you find very odd relationships where things occurring close to one another are quite distinct, whereas things quite far away are quite similar. And there's various genetic things that can cause cryptic lineages to exist, such as ploidy variation or genomic incompatibilities, which, again, if there's not resulting morphological differentiation, you can't pick those up without actually getting some genetic data. So going through a few examples here, this is just a comparison between two different closely related eucalypt species. So we've got white box, common species of inland woodlands through Southeast Australia. We've got blue box here, Barringana, which occurs in riparian areas in the southeast corner. Much more restricted than Eucalyptus albans. When we look at the isolation by distance relationship between these, which is just, this is genetic diversity between pairwise populations. This is geographic distance between them. It's a very slight relationship there with white box, but basically genes are moving completely freely right across this huge distribution. Didn't sample in South Australia. I suspect that would be different, but even into the Snowy River here, seeing no real genetic structuring, we see pretty much pendictive gene flow. Eucalyptus Barringana, very closely related, but a much stronger relationship between genetic divergence between sites and geographic distance. In fact, in this one, we actually see unexpected geographic structuring, and that we have two main lineages. One's down the south coast here, and the other's through Western Gippsland and over in Melbourne. So these populations over here, Western Melbourne, are actually more genetically similar to these populations in West Gippsland than the population in West Gippsland are to populations in East Gippsland here. Couldn't really pick that out without genetic data, could you? They look morphologically the same. So that's sort of the thing we're looking at. And then, of course, that informs conservation. None of these are threatened. Eucalyptus albans of used to replant, ecologically dominant, koala habitat, that sort of thing. Basically, it's showing it doesn't really matter where you get seed or to go plant. It's going to be pretty similar to what's there locally. Whereas Eucalyptus Barringana, which is used in restoration, grows in Western Sydney and stuff, councils planted out. It does matter. You don't want to be planting stuff down here up in Sydney, which has occurred. Examples of that, because you're planting a very different genotype to what's there naturally. Is it adapted to local conditions? Probably it's a eucalypt, but in other cases it may not be. And you may see that your restoration doesn't perform very well. Here's a cool example. I wasn't actually involved in this one, so I don't want to take the credit. This was other colleagues down at RISA. Ferrosphaler fitzgeraldii. So this is a gymnosperm that grows in waterfalls in the Blue Mountains. Very restricted to a few little waterfall areas. Genetically, found really strong clustering into two groups here, one of which is quite widespread through all the Western populations. One's restricted to just these Eastern populations. Bit of a mystery why it's structured like this until you map out the prevailing wind direction. So this is conifer wind pollinated. Prevailing wind direction perfectly matches those genetic populations. So they're being driven by the ability of pollen to disperse between these sites. It's important for management. It's Wentworth Falls over here, which is very genetically distinct. It's the largest population of it. The others are quite small. And so there's actually low genetic diversity in all of them. So the management recommendation here is that go pick up some seed from here, plant it over here, increase the genetic diversity in the largest population. These are not restricted due to habitat loss or anything. It's just habitat type waterfalls. Not exactly a lot of them around. So the largest area of habitats up at Wentworth Falls. So go bring some of this western genotype into the eastern genotype, increase that diversity. Hopefully you see population continuation in front of climate change and other ecological challenges. But yeah, very cool example where you get really neat mapping between wind direction and pollen flow basically. And here's Stemata triandra. These are the sort of ones that are ahead. Actually, this one's pretty easy. Took out the Dodonea. That gives me nightmares. Stemata triandra, so that's a grass, kangaroo grass, known to have varying ploidy levels between populations, potentially associated with climate, though sorry about that. Looked at it primarily in eastern New South Wales here. We find two distinct genetic lineages, blue one down south, the red one up north. They're very different from one another. So what you see here is an same IVD plot where it's genetic difference between population pairs, geographic difference between population pairs, but here they're colored by whether they belong to the same cluster or to different clusters. What you see here is this top row here. That's all the comparisons between populations in these two clusters. These middle ones here are all comparisons with these purple sites, which are genetically intermediate. We can then look at ploidy levels in a very proxy roundabout way, which I probably can't publish, to look at ploidy levels in these populations. And we find that both the red lineage and the blue lineage are either diploid or autopolyploid, as in they might be polyploid, but the copies of the genome are the same. Whereas all the stuff in purple is allopolyploid, and it contains one genome from the blue lineage and one genome from the red lineage. So we can get that from allele frequency data, our sequencing data. Basically, this is a tetraploid. You can see there's peaks at a quarter, half, 3/4 nearly. Whereas a diploid here or an autopolyploid has a nice normal distribution, and that's just related to if you have 4 copies of the genome, it's possible to have an allele only 1 copy and a different allele 3 others. Whereas if you're a diploid, well, you can only have an allele in 1 and not the other. I've been talking for way too long. Species reproduction, I might skip over a lot of that. That's pretty boring, other than to say people like to plant eucalypts, koala habitat. Koalas are good for one thing, that's for getting funding to look at eucalypts. Eucalypts like to outbreed. We say, how many children would you produce if the only people around you were your siblings? A lot of these plantations are probably, a lot of these restoration plantings are probably much more akin to a plantation than actual ecological restoration because they don't source genetically divergent seed. They're not going to see any self-sustaining populations resulting from it. Not great for the koala who has food for a few generations and then doesn't. Won't touch on that. This is probably more interesting stuff. An investment in genetic data can improve the cost effectiveness of other management actions. So looking at doing genetic study before you do any management actions to then inform what you're doing, including listing species, which we're working on. Fontania Herbarium from North Coast, New South Wales, known for 55 plants can mature in the year 2000. Genetic study was undertaken and it showed that very few of those are actually contributing to the seedlings. A little bit of guerrilla management happened. I don't think it was officially approved, but there was translocations between those sites, some very low cost intervention of translating a few trees. There's probably more Fontania area in this photo than there was in the year 2000. As soon as you bring in some different genotypes to a population, they start reproducing again. So what really was driving the species to extinction was just the lack of genetically divergent mates. So nice conservation outcome there. They count, it's up to more than 6,000 individuals now from the original 55. Grevillea hodgii, so this one's in Queensland, it's up Coochie Hill and Glasshouse Mountains. One of the most threatened plants in Australia, although I dispute that. I'd say it's one of the most naturally rare plants in Australia. Effectively, census size of 250 individuals. Effectively, the population size is 1. They're all genetically identical. They're not clonal, they're just so inbred, so many generations that they've purged all genetic variation from the population. You can sort of see here that 0.4 of the markers we looked at were polymorphic, i.e. had more than one allele, whereas for these other species, related species blood populations, you get up to about 20 to 30% polymorphism. So basically, this species has been really rare, restricted to Coochie Hills for a very long time, seeing no loss of genetic diversity or population in the face of European colonisation. Pretty good. However, it is probably very susceptible to any sort of disturbance event. Say Phytophthora gets in there and it's not resistant to Phytophthora, there's no variation in that population, you could see very quick population collapse. So here, probably what you want to do is maintain a small ex situ insurance population in case something like that does happen. But because there's so little variation, you don't need a very big ex situ population to maintain the genetic diversity. I think when I did the optimization of this, if you planted out 8 individuals, you capture pretty much 100% of the genetic diversity of this species. It's not very hard to find plant ground to grow 8 grevilleas in. Although you wouldn't want to grow more and then get a seed orchard going, but anyway. So final thoughts. Can we gather genetic data for all threatened plant species? Well, this is what a typical study looks like nowadays. Develop a sampling strategy, that's pretty easy, do that, it's routine. You sample prep and sequencing, that's all done by robots down in Canberra now. It's no lab work, I haven't set foot in a lab for a long time. Then you do the data analysis, interpretation and reporting, it's all done in standard pipelines now, standard format, that's pretty easy. And then you can guide on-ground management activities. So this is just going through end to start. We've developed all these pipelines just to think about what you need, plan a single sampling event, think about what your questions are so you can address all those questions with one sampling event, and then use standard pipelines. A lot of it's R, but there's standalone programs as well. Just run simple scripts that just spit out a PDF of everything you need to know about the species, basically. Sequencing's cheap now. Nice genomic revolution driven primarily by human genomics, biomedicine. It's great. But flow-on effects are good for us working in natural systems. And in fact, but their field work is more costly than actually doing the genetic sequencing nowadays. And is the biggest time sink. So yeah, that's where we're stuck. Trying to get this done for all threatened species is the sampling. There's no way around it. Working on a threatened species, you have to go out to where the species is, collect it, bring it back, and that can be very hard when it's, as I've been dealing with the last few weeks, growing on the edge of a cliff up in Lamington. That's a difficult prospect. And yeah, the costs are far from prohibitive. So this is some work colleagues, again, down in New South Wales did, looking at how much money goes towards genetic studies under the Saving Our Species program down there, which is really incorporated genetic study into the management pipeline. And on average, it's about less than 9% of the cost of a management project goes towards that genetic study. That genetic study informs all the rest of the management actions and helps to make sure they're as cost effective as possible. So can we gather genetic data for all threats? Yeah, let's aim high. We can do it. Not that hard. Do things like get economies of scale. You have standardized pipelines, data generation, sampling techniques. We're sort of implementing all that at the moment. Makes things very easy. It's relatively low skilled now. You were a student starting with me, I wouldn't even really expect you to be able to code anything in R. I could give you obviously you'd want to learn as a student, but I could give you the scripts to run, and then it's just about you interpreting those results. People are getting a lot more interested, so there's a lot more funding around. And by centralizing things, we can sort of pool that money, get benefits. And yeah, we don't need to know about everything. We just need to know the important things about everything. But yeah, there's a lot more work to be done than hands to do it. And so very open to collaborative opportunities to look at Queensland's flora from a genetic standpoint. And yeah, just finished with the acknowledgements.
Questions?
Thanks for the talk. What's the approximate time frame between discovering a species should be lumped together or split up and get that published and then get that a change in the listings?
Depends would be the answer. If there's somebody to write the paper, that can be a very quick paper to write, publish, go through publication to one of the Herbarium journals. Small change like that, probably where you go, they're pretty quick turnaround. So you can have that paper done in a couple of months. The changes in listing, that's going to vary a lot more and be more complicated because any change to taxonomy would have to be accepted by the state's botanical authority, which here in Queensland is the Queensland Herbarium. So they have to accept that change to the official statewide arbiter of taxonomy in Queensland. Once they're accepted, then it has to go through the whole process with its listing. So it can be a long process. I know again, New South Wales, I think Banksia Vincentius just coming off around now, the listing. So that was a whole story. I'm not sure if anyone's familiar with that, but basically Woolworths wanted to build a car park. There was brand new Grevillea, Banksia discovered there, which turns out had come from somebody's garden, and was just not a local. But that got listed and has taken something years, 5 years or something, was when it was proved not to be a distinct thing. So it's not a quick process, but you know, we also want to build confidence. Some people, I've had comments on the lines of, you know, is that really a good conservation outcome? You're removing protection from these species, which removes protection from the land they're on. But it's also about developing that trust with stakeholders that what you're actually saying, what you're conserving is based on empirical data. So, you know, acknowledging when you're wrong and taking stuff off is just as important as well, so that, you know, you're building that trust with people that what you're saying is actually based on empirical data.
I was interested in the first two examples that you gave, and I like study movement of animals, so use small genetics words for me. But basically, I think my understanding was the first point you were like, yeah, it's morphologically distinct on the headland, but it's genetically the same, so whatever, lump it in.
Yep.
Then the second point you said, like, there was one of the subspecies that was kind of morphologically distinct, so you kept it even though it was genetically the same. So could you say that?
It was genetically the same, as in not highly divergent from the other species, but unlike the headland. So the headland population, the real, I guess, nail in the coffin was that each headland population is most closely related to the local non-headland population. So even headland stuff, it's not forming a single image. As in the eucalypt example of Canodolensis, it was actually more divergent than expected from the local rubida that grew to the bottom of the mountain. So, you know, not divergent enough to be a separate species and probably still having gene flow, but it is a different habitat up the top of the mountain there. And it is somewhat genetic divergent to the start of the bottom of the mountain, which is where the subspecies classification sort of comes in.
Whereas in theory, you'd expect if you just took one of those land plants and just whacked it on the headland, it would develop a short term track.
I mean, the main difference is the growth form, really. It's some other stuff, like leaf size and stuff, but that's all going to be heavily impacted by the wind coming off. I guess that's where it didn't really touch on there, but geographic patterning of genetic structure is very informative for taxonomy as well. If you've got images that are overlapping and not experiencing gene flow, well, then they're going to be different species. Whereas it's very difficult when you have geographically isolated species that are not experiencing gene flow. That's where it's, don't know which way to go or something.
Okay.
got a question from Christine Hosking in the chat. So thanks for an interesting talk. Can frequent management burning affect genetic diversity?
Interesting question. Depends on your species. If it's not something I've looked at directly, but I guess if you've got some, I think of an example of where a species would be selectively resistant to fire.
Thanks, Ken.
How fire influences reproduction would be critical.
Yeah, but it would only impact it if there's differential responses based on genotype. If the entire species is responding in the same way, you could lose genetic diversity or you could lose population. Something that's not going to affect genetic diversity per se, it's just going to affect the entire population. If you've got a species where, I don't know, some genotypes resprout and others, which does happen in Banksias, others will only reproduce by seed, then, you know, fire regime may change which genotype is favoured in a habitat or something. It's going to be probably less uncommon. Fire's pretty all or nothing sort of disturbance. Things that are going to have much more impact are disease, because there's going to be a lot of variation in disease resistance between genotypes, that sort of thing. Those sort of disturbances probably more going to have an effect on the genetic diversity than burning in and of itself.
Thank you, Patrick. Good to see you.
Yes.
Well, like ponytail. It's not it's just. So there's obviously a very dominant paradigm in restoration, and local provenance is better.
Yeah.
And I've dealt with lots and lots of community groups over the years where they take that view very, very strictly to be like within a kilometre.
Yes.
And all of Suzanne Prober, Martin Breed have said they went and looked at the genetic diversity in restoration plantations, and I call them plantations like you because that's what they are. And much lower genetic diversity in those plantings than in a sort of adjacent intact habitat. Maurizio's work showing that, you're showing that. Is that message getting out?
Yeah, people are picking up on it. I guess the tricky thing with that situation is if you go back 20 or 30 years, the people who were circulating that idea of local is best are the same people who are now pushing back against it in population genetics and that sort of thing. You know, a couple of decades ago, it was the idea that things are locally adapted, that sort of thing, and that's part of where that came from. But yeah, no, the opposite is getting traction, both from theoretical and practical standpoint. People sort of doing work with, well, anymore, but when I was down in New South Wales doing work with Taronga Zoo and nurseries doing koala habitat restoration out in Northern Tablelands, nurseries were very interested in diversifying the seed sources they've got available to sell to people. So that's a value add. They can sell that seed for a high cost, you know, say, oh, here's our baseline seed, here's our highly diverse seed, or whatever, so they can do that. And then there's also the practical standpoint of it is unique. It's hard to get seed to do restoration. The wider area you can source your seed from, the easier it is to source seed. So there is that side of things where it's actually people are on board because it makes their life easier. But yeah, I think the momentum is definitely growing there. That local is best is not true. So we published some work basically saying that the only way to really understand where to go source your seed is to actually do a genetic study of that species. You can use proxies, that sort of thing, but those proxies. So we can look at landscape mapping and community type mapping, that sort of thing. Generally, they have a very poor relationship to actual genetic patterns and species, which vary very heavily between species. Yeah. I've had conversations with rangers in national parks down near Sydney who were very annoyed they couldn't source seed from their particular ecological community. And if anyone's familiar with ecological community mapping in New South Wales, it's a mess, and you get tiny plots where there's one tree or one community anyway. And so, you know, they're very willing to say, okay, go source seed further because it makes my life easier.
I have a question about the genetic data. So I presume you would want functional diversity and not just genetic diversity.
So all neutral data. Basically, there is that idea that you want some sort of adaptive signal, that sort of thing. But then a lot of work has gone and said, okay, what's the signal on the adaptive genes versus what's the signal on neutral genes, and 95% of the time they give the exact same signal. So when you're looking, we're not particularly interested in individual adaptive traits because we're dealing with a very large number of species, very large things. We can't be doing a 20-year greenhouse study for every species. We need answers within a year, that sort of thing. 6 months all the time. So we're just using neutral data, just looking at what is the actual pattern of gene flow, because that neutral genetic data is going to be flowing across the landscape, basically in tandem with any adaptive potential. And we also find that really plants are not that closely adapted to that in the first place.
I'm just going to make a comment, kind of invite a bit of discussion. Yeah, so there's this great tradition of sort of morphological botanical taxonomy where people look down microscopes and they're absolute experts at discriminating micro characters, plants, to build this thing we call plant taxonomy and all the names we're used to. And the language for the things we love that we use to talk about these, the fabulous diversity of botanical life. And then you guys come along and just put bomb into the whole thing, basically. And demonstrate that things that are quite distantly related can, through convergent evolution, end up looking the same, and things that are identical can be, through geography and isolation, can be completely different. How do you feel about that? So,
look, for all there is here, when taxonomy doesn't line up, and 9 times out of 10, it lines up. The existing taxonomy-based morphology lines with genetic data. It's not like we're revising everything in every example. And it's the exceptions that get shown here. And it is heavily independent on what group you're in. But yeah, it's also the side of things that what good is a taxonomy if you can't discriminate the species in the field. So that morphology does need to be included.
We'll more often discriminate species based on geography than we used to, won't we? Because genetics will show that these populations are isolated, and then we can talk about the Blue Mountains ding dang dong versus the Queensland ding dang dong.
Yeah. Comes back to what I was saying earlier, where we're not really decided on how to approach that yet, where in situations like that, where you've got genetic structuring between geographically distinct groups, the genetic data isn't actually that informative whether they should be species or not. And so if you find genetically divergent populations or a thing, I wouldn't be calling them different species until you go find a morphological trait that you can separate them by. It's mainly in those systems where you have overlapping geographical distributions where things are a lot more cut and dry. Yeah, that sort of goes to your point.
Yeah. I mean, yeah, it's hard to argue against the fact that we got 40,000 characters instead of 10, right? Yeah. I hear you. But my misgiving is that the whole agenda of botany and, yeah, building a taxonomy, which is about describing the diversity, it's kind of hijacked by the geneticists. The rest of us can't do anything unless we're totally beholden to you to get these pathways to the species. And then we put the show back together again.
It sounds like really bad control issue.
Yeah. Take the man's office then.
Yeah, I mean, I find the whole thing so fascinating. Right?
Yeah.
I'm not really aggrieved. Yeah, I've got a million questions to ask you about individual examples you put in there. But that Grevillea hodgii is a good one for local people. It grows on these hills back at the sunny coast. Anyone can go and have a look at it. It's and isn't that thing like widely planted in gardens? So like one of the cultivars of Grevillea. My mate runs a nursery and he hides the Grevilleas back at the nurseries because he wants people to buy the more interesting stuff, and they find their way to the Grevilleas and buy them anyway.
Supposedly,
isn't that one of those things? Yeah.
I don't know. You know, after a few of those cultivars in there, and none of them, they're all Whiteana hybrids, so I'm not sure whether that's actually true or not.
Anyway, there's still a narrow leaf Grevillea grass there, and then people are planting a related species in the gardens at the bottom of the hill.
Yeah.
And there's hybrids between them.
Yeah, well,
and you're painting this picture that it's got zero genetic diversity, this rare thing. Could it be that the bit of hybridisation from garden escapes is a good thing?
Yeah, actually having a workshop with the Sunny Coast Council this afternoon where we're going to discuss stuff like that. But yeah, one of the main concerns with the Hodgii is that Borrelia banksii, which is a close relative, it's from about Maryborough North, it's a native distribution theoretically, but it's really common naturalised escape cultivation down here. And so there were stands of it on Kuchen, and they were finding hybrids between that and Hodgii. We genotyped of them, turns out, yeah, they are hybrids, whatever, they're able to pick them morphologically. And then Parks went through and cut out all the Banksii and all the Hodgii, all the hybrids. Yeah, I am left thinking like, that probably wasn't adaptive for that species. It probably was helping conserve it. But yeah, it's kind of hard to explain that to land managers that sort of by losing the integrity of your species, you're probably helping conserve it. The other interesting thing about that is it's growing in Australia Zoo. They have it planted there. So chances are, even if the wild populations go extinct, there's already an insurance population that's there by accident. So who knows?
Yeah, love it or leave it. I think, yeah, that's a good example of where you have to accept that a foundation of genetics is pretty useful, isn't it?
And then
conservation work
comes back to the question I'm always left thinking about is we have a very poor conceptualization of what's a threatened species versus what's a rare species. I'd say Borrelia Hodgii is a very different case to something that's affected by murder rust and lost a lot of population, that sort of thing. Borrelia Hodgii is really a population on its way to extinction in the first place, and the human impacts are not really the cause of that. But it is so small, meets the criteria to be critically endangered, it gets funding, that sort of thing. There is that aspect of what is a threatened species.
Yeah, well, that Fontaine is a good example, isn't it? It's a rare rainforest tree. How many plants are there? About 15 or something. Someone discovered.
There was any mature.
It's got hardly any genetic diversity, doesn't know how to reproduce anymore. Isn't this a species that we should just let go extinct? Sort of philosophically, it's like run out of steam, isn't it?
And how important is it in the ecosystem as a whole? We're dealing with current rules and regulations, so we're going to have it.
Yeah, we shouldn't be able to help ourselves, I reckon. Extinction's pretty profound. We're trying to stop that, I reckon.
Yeah, and there's so many lines.
Yeah.
You want a clear cut one, just to prevent it. I could argue every case.
Yeah.
Any more questions? I reckon it's great, Patrick. It's all really exciting stuff, and it's great to have you here in Brisbane, and hopefully we can get some of those processes fulfilled through collaboration with UQ and beyond. Thanks a lot.
I'll send you a list of my favourite things that I want done.
Is there going to be money attached? Because no money is happening.
And I'll get to come in and do all the field work.
Oh, well, if you do the field work, there's actually a chance it might happen. But
yeah, good.
awesome. Thanks.